Hello, my name is Tim Tickle. I'm here to talk to you a little bit about how I used AWS Health Omics to process and analyze short read data. To give you a little bit of context about the data I was working with, imagine, for instance, you wanted to measure DNA in a sample. And so in this case here, we have samples and it goes through a sequencer. What you'll get out of that a lot of times are these short sequences of DNA. And so you have to make sense of all these short reads of DNA. And what one can do is, for instance, get a reference genome and then take that reference genome and align the reads to where they originally came from. And so in this case here, the reference genome is in green and the reads are in gray. And most of them don't have letters. But in this case here, for instance, there are a whole bunch of letters. And what that's indicating is the reference genome at this location had this DNA nucleotide, so A. And in the samples, though, they had a whole bunch of C's. And so this represents potentially a mutation, likely a mutation. And to be able to understand that, you can do downstream analyses, or you can also look at annotations. For instance, here we have an annotation showing where and what chromosome this mutation was from. And more to the point, ClinVar, for instance, indicating these locations can have mutations that, if they are there, could be pathogenic. And so this case here, we might know this variant or this mutation is from this chromosome and potentially is pathogenic. So how does it look like to do that kind of analysis in health omics? Well, firstly, your samples are turned into sequence stores, your reference genome is turned into a reference store, and both of those are combined together into a workflow that processes those samples. The results of VCF files that hold your mutations in those VCF files are put in the S3 buckets. Your VCF outputs are turned into a variant store, and your ClinVar annotations, for instance, are turned to an annotation store, and both of those can be queried in Athena, which I can show you right now. So we're currently in Athena, and there's a couple tables that we're working with. This first query is just showing sample data in the actual VCF files that we had, or the variant store. The second one is showing annotation data from ClinVar. This third one was a query that I put together just to see how many mutations in the samples came from different chromosomes. And then here, lastly, is a more interesting query. And so for this query, what we're looking at is if you take all the different variants in the sample and you join them with the locations in ClinVar that are considered to be likely pathogenic, what are the results? And so we get here 9 different mutations that are considered likely pathogenic. And that query uses both tables combined together. And thank you so much for your time.